Hello everyone, Yan and Gina. Welcome to my channel. And this video is going to discuss the first chapter of AP Biology, which is Chemistry of Life. So, in this video, this is about the content, and we will introduce this formula parts. The first is about the water molecule, and we will talk about the structure, also properties, and we will going to discuss 3 different chemical bonds, and also the makeup of macromolecules, and a brief introduction of common macromolecules. So, and the first part is about the water molecule structure. We can see that this is about one of the water molecule, and it really looks like a Mickey hat. The largest part, the right one, is the oxygen atom, and the white part, which is 2 white parts, are the hydrogen atom, and they are like a triangle, and yes, the characteristics of the water molecule. So the first one, they have 2 characteristics, which are the cohesion and adhesion. Cohesion refers to the attraction of molecules for other molecules the same kind, and water molecules have strong cohesive forces, thanks to their ability to form hydrogen bonds with one another, and the cohesive forces are responsible for surface tension. This is a phenomenon that results in the tendency of a liquid surface to resist rupture when placed under tension or stress. Water molecules at the surface will form hydrogen bonds with their neighbor molecules, just like the water molecules deeper within the liquid. And another one is adhesion. Water likes to stick to itself, but under some circumstances, actually it prefers to stick to other types of molecules. Adhesion is the attraction molecules of one kind for molecules of a different kind, and it can quite strong for water, especially with other molecules bearing positive or negative charges. And this is an important example for the adhesion, which is it can enable the water to climb upwards through the plant's xylem. This upward motion is against gravity, known as capillary action, and it depends on the attraction between water molecules and maybe the glass walls of the tube, as well as interactions between water molecules. So in biology, there are 5 properties in water, and we need to remember them. The first one is the high specific heat. It refers to the amount of heat needed to change the temperature of 1 gram of a substance by 1 centigrade. Water's specific heat is very high because of their hydrogen bonds that hold the water molecules together. So this property is excellent for helping them to maintain a constant internal body. The second one is that it is the excellent solvent. Water is often referred to as the universal solvent. This means that it is great at dissolving other materials as solids. And the third one is the high heat of vaporization. It refers to the amount of heat needed to convert a liquid to a gas. Due to the many hydrogen bonds with the water molecules, which hold water molecules together, the heat of vaporization is very high for water. And cohesive and adhesive, which talked about in the last one, is can be applied in the transpiration plants. Yeah, it is polarity, water molecules stick to each other and to other poly molecules. This property allows transpiration, the movement of water through the xylem of plants, to move water from the ground against the gravity through the plant and eventually out in the air. And the last one is their different density in the liquid form and the and their ice form, because the volume of water contracts as it cools down, but unlike other materials, the volume of water begins to expand after it cools to temperature below 4 degrees Celsius, and this causes ice to be less dense than liquid water. This means that ice floats on liquid water. So this part is about chemical bonds. We are going to learn 3 different kinds of bonds, which are covalent bonds, ionic bonds, and hydrogen bonds. Covalent bonds is a kind of strong bond because it results from the sharing of valence electrons between atoms, and those atoms held together by covalent bonds are called molecules. And the second one is about the ionic bonds. It occurs from the metal and non-metal atoms. Compounds held together by ionic bonds are called salts, like the sodium chloride. It has the ionic bonds. And this last part is about the hydrogen bonds. Hydrogen bonds is fairly weak compared to covalent bond and ionic bonds, and it is kind of the intermolecular attractions. So this part is about the makeup of macromolecules. We need to learn the identification of polymer and monomer. Polymer, which are structures made of repeating smaller units called monomers, like carbohydrates, lipids, proteins, and nucleic acids. These are all the polymers. But their monomers is that the units that compose them, and the hydrolysis refers to that the reaction from polymer to monomer, like the monomer of the protein is the amino acid, and when an amino acid and when the protein convert to amino acid, it needs hydrolysis. The protein needs to add water to split in lots of the amino acids, and the hydrogen synthesis or condensation reaction refers to the reaction from the monomer to polymer, like when amino acids need to convert to proteins, and need the hydrogen synthesis. This refers a covalent bond forms between two monomers, releasing water. So this is an example of the hydrogen synthesis, which I mentioned in the last one. This is the structure of an amino acid, and when it joins the other amino acid, the carboxyl group and this one will composed with this one in another amino acid, and will form a water molecule to form the peptide bond, and lots of these amino acids will form the chain, and eventually they become the protein. So this is the brief introduction of common macromolecules, and we need to study 4 different macromolecules. The first one is about the nucleic acid. Its monomer is the nucleotide. Its function is to store genetic information. We are familiar with DNA or RNA, and the nucleotide is composed of phosphate group, a sugar, and a nitrogenous base. And this is the structure of DNA. DNA is the polymer of many of the nucleotides, and DNA is there are different kinds of nitrogenous base, and in DNA there are ATGC, and in RNA there are AUGC. Hydrogen bonds occur only between complementary pairs, between C and G or A and T. When C connects with G, they have 3 hydrogen bonds, while when A pairs with T, with double hydrogen bonds. The second one is about the protein. So protein, or we can consider there are 4 parts in an amino acid. The central is a carbon atom, and there must be a hydrogen atom. Also, in the carbon atom size, one said about the amino group, and the other said is about carboxyl group, and the last part is about the R group. Well, will compose different R group because they're just a general form of the amino acid. And in protein, the elements composed protein are carbon, hydrogen, oxygen, nitrogen, and sulfide. Protein have lots of functions. They can repair our injury, or they can act as hormones or signals, and the very important point in this part is about the levels of the protein structure. There are 4 levels of the protein structure. The first is the primary structure. It refers to the sequence of amino acids. And in biology, a mutation in DNA can change the primary structure of protein, and this change can vastly change the overall shape of protein and cause it to lose its function. Also, a change in the primary structure in biology, the other 3 structure will also change, and the second one is the secondary structure. It refers to hydrogen bonding between polar amino acids, and it includes the alpha helix and the beta pleticate. And as their name, the alpha helix is the helix, and the beta sheet is about the like the parallel structure. And the tertiary structure, is partially determined by ionic interactions between certain R groups and by hydrophobic interactions between certain R groups. And in tertiary structure, it appears disulfide bridges, like this one. And the last one is the quaternary structure, the most complicated structure. It refers to two or more polypeptides are woven together to form the overall structure of protein. Like this is the example of hemoglobin. It has four polypeptides woven together to form the hemoglobin. So this is the summary of four levels of protein structure. In FRQ, sometimes you are asked to explain what is the primary structure of protein or what is the secondary structure of protein. So maybe you need to remember them. And we are also talk about the denaturation of protein. It refers to unraveling or unfolding of a protein or enzyme causing it not to function. And it can be caused by the change in pH, the salt concentration change, and the increasing temperature, like about the 37 degrees. The denaturation usually disrupts the secondary and tertiary levels of protein structure by affecting the hydrogen bonds or the sulfur bonds, because they're responsible for stabilizing the structure. And in P-biology, as I mentioned, when this primary structure changes, the secondary, tertiary, and quaternary structures all change. This is very important. It usually appears in the multiple choice. And next macromolecule we're going to study is a carbohydrate. It serves as sources of quick energy and building materials. So carbohydrates very important in our body. And most carbohydrate names end in the letters OST, like lactose, fructose. And carbohydrate can be classified as monosaccharide, disaccharide, and polysaccharides. Monosaccharide is the simple form the sugar. Usually it is a composes glucose, fructose, and disaccharide. Usually the sucrose, lactose, and maltose. And polysaccharide, there are four polysaccharides in biology which are very important. The first one is starch. It usually used as an energy storage molecule in plants. And next one is glycogen, used as an energy storage molecule in animals. The next one is cellulose. It's a part of structural component plant cell walls. And the next one, chitin. It is in the exoskeleton of some insects and also fungal cell walls. The next part is about the lipid. There are two main parts of the lipid. The first one is the fatty acid. It's the long chain of carbon and hydrogen atoms. And another part is 3-carbon glycerol molecule. And this one, it will hold the whole molecule together. So we can consider this is a height, and this part is about its tail. And the lipid can be classified as saturated and unsaturated. Saturated, there is no carbon double or triple bonds in these chains. This one, all bonds between carbon and 10 carbon are a single bond. So they are solid at room temperature and they are usually associated with animals. And this is uh, and this is what the unsaturated lipid look like. Because there is double bond between the carbon atoms, so it could be broken. So it is fluid at the room temperature, and they are usually associated with plants. In pea biology, there are three types of lipids. The first one is phospholipids. Phospholipids is found in the cell membrane, like this diphospholipid bilayer. And the second one is steroid. It's a kind of hormone, and we'll talk about later. And the third one is the cholesterol. It's a combination of steroid and alcohol, and it is also found in the cell membrane, and this main function is to increase fluidity. So, like this phospholipid, this is the hydrophilic head, which means that it can interact with water molecules, and it still is hydrophobic, which means it cannot interact with water molecules. And those are the practices. And you can push the stop button to finish these practices. And this is the second video. Hope you can like my channel and hope you can get a great score in this course. Thank you. Bye bye.